Hi, I'm Mrs. Schramm, and today we're going to learn how to do a bacterial transformation. So we're going to run through this really quick to give you some ideas on how to do this lab. First of all, you're going to need to have the following things. Make sure you know where the hot water bath is in your room. Make sure you have an ice cup, a clean set of tips, a waste container that we're going to be putting E. coli waste, so make sure not to touch in there. And then you're going to need your P20 and P200 pipettes, and you're going to have a tube rack. Your teacher will later come by with some things called spreaders for spreading the bacteria. So today, what we're going to be doing is we are going to be giving you some live E. coli cells. These E. coli cells are what we call competent cells, so they'll be labeled CC. It's important that they stay cold on ice because the cold temperature is creating these adhesion zones in which we can try to sneak a new piece of DNA into the cell. So it's critical that everything stays cold. So pay attention. So we're going to start you guys off by giving you guys two empty tubes. You are going to label them P plus and P minus. You're going to put these tubes in your ice cup. The tube has to stay cold because the P plus and P minus tubes are both going to be getting some competent cells. The teacher will come by with a tube labeled CC for competent cells, and that has to stay on ice. Notice I was holding it by the lid because temperature is critical. I'm going to start off by taking my P200 pipette. I'm going to set it to 50 microliters, and I'm going to get a clean tip. That clean tip is going to be used with E. coli cells. So I'm going to start off by taking my competent cells. I'm going to open it carefully, holding the tube by the tip. By the rim. Okay. I'm going to press down to the first stop. I'm going to mix up and down, so that I can mix the cells because cells settle to the bottom. Then I'm going to make sure that I've pressed down to the first stop and that I suck up 50 microliters of the solution. I'm going to put that into my P minus tube. I'm going to try not to touch the bottom because I don't want to warm up the tube. I'm going to do the same thing again. I'm going to pipette up 50 microliters of cells. I'm going to put it into my P plus tube, and I'm holding it at the top so that I don't warm it up. I'm going to eject the tip because it had E. coli on it. Next, P minus, that's going to be a negative control group. So what I'm going to do is I'm going to be adding 10 microliters of water. I'm going to set this one down because that's not the pipette that I need. I need to switch to my P20 and have it set to 10. So that should look like it says 100 because the decimal is after the second zero, or the first zero here. I'm going to get a new tip. I'm set to 10. I'm going to pipette 10 microliters of water, and I'm going to put that into my P minus tube. I'm going to make sure that I mix the water in, pumping up and down, and then pipetting out to the second stop to make sure my tip is empty. Okay. I'm going to make sure that I keep this on ice. Notice I did not hold the bottom of the tube. Okay. Let's do the same thing again. This time, I have a clean tip, but I'm now going to be using my DNA. In this tube that says RP, that's my recombinant plasmid. That's what I want to sneak into the bacteria. So I'm going to get 10 microliters. I'm going to get my P plus tube. P plus means yes, plasmid. It has a plasmid. I'm going to add the plasmid and pump up and down a few times to mix it. And I'm going to put the lid on. And then I'm going to eject my tip and I'm going to start a timer. My timer needs to be started for 15 minutes. Okay? My 15-minute timer is going. I have 15 minutes that the P plus tube has the cells and the DNA in the same tube so they can be near each other, okay, for 15 minutes. While you're waiting for that 15 minutes to be up, you're going to prepare your Petri dishes. These plates, or Petri dishes, whichever you like to call them, I usually call them bacterial plates, each have a line on them. This has 1 line, 2 lines, 3 lines. You're going to do them in that order, first, second, and third. Your first Petri dish has 1 ingredient, and you're going to draw a line down the bottom of your Petri dish. So there's the agar layer. Draw the line down the bottom, P minus, P plus. Okay, that's what you're going to write on there. Next one, on the second Petri dish, write the same thing, P minus and P plus. On the third Petri dish, you're not going to write a P minus. You're just going to write P plus, and this Petri dish is going to grow lots of transformed bacteria. So we don't want to waste any of our negative control space on this Petri dish. So let's pretend that our 15-minute timer went off. Okay? What we're going to do next is we're going to carry this ice bucket over to the hot water bath. Make sure the lid is off of the hot water bath. Make sure your partner has changed this to stopwatch. And what you're going to do is one partner is going to get the tubes ready, the P plus and P minus tubes, so they're both in one hand, okay, only holding it by the lids. And the other lab partner is going to say, ready, go, and start the timer. And that lab partner is going to put the tubes on hot water. This water is only 42 degrees Celsius. It's about bath temperature. It's not going to burn you. You need to hold this in here for exactly 45 seconds at 42 degrees Celsius. As soon as that 45 seconds is up, back on ice. Timer restarted, keep it on ice for 1 minute, okay? After that 1 minute has happened, the bacterial transformation just took place here in this heat shock. We don't need the adhesion zones to stay opened anymore, so after 1 minute on ice, we can transfer these tubes to the green rack, or whichever color your rack is. And then we need to make sure that these cells are happy. So we're going to get our P200 pipette set to 150. We're going to give it something to help the cells recover, because the cells have been through a lot. We've heated them, we've cooled them, we've heated them and cooled them. We're going to now give them 150 microliters of chicken soup for cells. That's what I like to call LB broth. I'm going to add 150 microliters to my P plus tube. I'm going to pump up and down to mix it in. I'm just pumping up and down once. I'm going to get a new tip again. Remember to change your tips so you don't cross-contaminate your samples. I'm going to get more LB broth, another 150 microliters. I'm going to put it into the P minus tube, pump it up and down once, okay? Now I need to plate my bacteria. So the teacher will come around with these blue spreaders. We're going to get a clean tip, and we're going to start off with our P minus. On 2 Petri dishes, I need to make sure that the agar side is touching the table. So they should be right side up with the lid on top. I'm going to take my P minus tube. I'm going to suck up 50 microliters. I'm holding the wrong pipette. Hold on a second. Okay, 50 microliters. I want to make sure I have a clean tip. And I'm going to transfer 50 microliters onto my P minus side. Notice I'm only opening the Petri dish lid a tiny bit, just enough to squirt this on there. Want to make sure that the lid is up. Okay, there we go. Now I'm done with that. The teacher is going to give you a spreader. You're going to take this spreader, make sure not to use it like a shovel. Spread gently the P minus. Spread gently on the P minus side only to move the bacteria around so it takes up half the Petri dish. Dispose of your spreader there. Make sure to get a new tip. And then get your P plus tube. Here's my P plus, my transformed bacteria. It's genetically modified now to contain new DNA. I'm pipetting 50 microliters onto the P plus side of my Petri dish. My last Petri dish has only a P plus side. So I'm going to pipette 50 microliters twice for a total of 200 microliters. 50 plus 50 is not 200. For a total of 100 microliters is what I meant to say. So I'm pipetting 50 microliters twice onto this last Petri dish. And now I'm going to need another spreader from my teacher. I'm going to spread the P plus side. Notice I'm starting on the first Petri dish. This Petri dish has one ingredient, LB for nutrients, and that's it. This Petri dish has LB, but it also has an antibiotic named ampicillin. If I was to start spreading on this Petri dish and go this way, I would transfer that second ingredient onto my first Petri dish, and I would cross-contaminate things. So it's really important that you go from Petri dish 1 to Petri dish 2, and finally at the end to Petri dish 3, because there's a third ingredient on this Petri dish, which is a sugar called arabinose. That's going to help turn on the gene to make sure it gets expressed correctly. So I've spread everything. After I've spread everything, I want these to sit upright for 5 minutes. But at the end of that 5 minutes, I want to take a piece of tape, wrap a piece of tape around all 3 Petri dishes, and then stick these into the incubator upside down. I'm gonna let those sit till next class, and then we're gonna see if we were able to transform our bacteria. Good luck with the lab, guys.